All right, so he's a physician, he's a philanthropist, he's an Emmy-nominated film producer, and a longtime supporter of LABEST. Please welcome Dr. Eric Esrailian, Division Chief of Digestive Diseases at the David Geffen School of Medicine at UCLA, to lead a discussion on microbiome science at UCLA. Welcome.
Thank you so much, Judy, and it's an honor to be here, and thank you all for participating. You know, this is an exciting time of the year, both for UCLA and obviously for all of the life sciences, biosciences community, and bringing people to LABEST is exciting. So I just want to let you know we have a very esteemed panel, much more esteemed than me. So Dr. Gary Michelson, Dr. Elaine Hsiao, Dr. Greg Donaldson, Dr. Jonathan Jacobs will be on our panel, and we will be discussing microbiome, but also talking about the intersection between our Goodman-Luskin Microbiome Center at UCLA and the California Institute for Immunology and Immunotherapy. And Dr. Gary Michelson is an overlap of both these topics, so I'm really excited to have them here, and thank you. And I'm excited about the golden ticket as well, for those of you who like Willy Wonka and the Charlie and the Chocolate Factory. So just by brief background, and I know you've heard about the California Institute for Immunology and Immunotherapy in bits and pieces probably over the last couple of days, but a few years ago, the gentleman who named this amazing conference center, Meyer Luskin, had talked to me and Chancellor Jean Block at the time about supporting something innovative in science that already had strength at UCLA that we could take to another level, and he brought Mr. Donald Goodman and his family to the project, and we ultimately named the center the Goodman-Luskin Microbiome Center, which is really now the core of all microbiome science at UCLA, and it pulls from multiple different disciplines and skill sets, and Dr. Hsiao will be talking a little bit about that. But you know, the establishment of this center right on the, you know, kind of the cusp of the launch of the California Institute for Immunology and Immunotherapy also was, you know, really a kind of converging of two huge projects that we wanted to kind of get out into the world, but they're extremely complementary in terms of some of the projects that you're going to be hearing about today. And I think the partnership between the California Institute for Immunology and Immunotherapy and UCLA and other academic institutions in the state of California is what we're trying to kind of talk about and build on for years to come, and launching some initial projects at UCLA through the Goodman-Luskin Microbiome Center is what brings us here today. So again, it's an honor for me to be a part of it, to be a co-founder with Dr. Michelson and Meyer Luskin and Sean Parker and Ari Beldegren and am I missing anyone? Mike Milken. So Mike isn't here, but you've seen a lot of the other participants here. So I'm going to start with talking to Dr. Hsiao and get a sense from you, Elaine, in terms of what is it about UCLA that made it a special institution to bring together all of these great scientists for the purpose of creating this Goodman-Luskin Microbiome Center. What was unique about UCLA, and as the leader of this center, what makes you feel like this is the best place to have such a center?
Yeah, sure. For the microbiome, it was really only just sequenced in 2012. And so it's been relatively early days. What people realized over the years is that microbiome touches on so many different areas. Microbiome in medicine, structural biology using computational tools, the ecology of the community, and it touches on virtually every discipline. And to truly make advances in the field, you need expertise across these disciplines. And so I think it's a bit underestimated, but UCLA having top-notch scientists and clinicians, and also being all in the same place, getting all the right people in the same room is what I think was a true opportunity here. In other centers, they do great work, but it's oftentimes built around like a few labs that are really excellent in the field and have their unique perspective. Here, I think we can really, truly do multidisciplinary research that will advance the field.
And can you talk a little bit about how you started to pull together colleagues who had an interest. I mean, obviously you had some collaborative relationships already at UCLA through your own specific research, but there's a transition from being even a prolific scientist who has a lab and is publishing and doing amazing work and trying to lead a center, even if you have resources. There's a shift, and I've noticed even your leadership has really been thriving and growing as you transition from being like this very well-known microbiome scientist to somebody who's actually now leading a center. So can you tell us a little bit about that process?
Yeah, sure. I think this is a second point that I think feeds into why UCLA was especially poised. Just thinking back of when I joined UCLA, I remember my first impression was that, wow, everybody responds very quickly and very generously to my requests. And so I've been, at many different places, and that's not always the case. So I think the level of collegiality, no egos for the most part, has really facilitated this culture for being able to contribute to a shared mission. And so pulling together people from the clinic, from technology side, from basic science side, was everyone says yes, basically. And so that was really, I think, another thing that makes UCLA unique in this regard is just having good people and collegial people.
Amazing. And then, before we kind of talk to some of our other speakers, I wanted you to also describe the process of recruitment, because Dr. Donaldson wasn't here when we started talking about the center concept, and then you quickly identified him as somebody who could add a unique skill set. And can you talk a little bit about the process of recruiting to bring talent to UCLA, to the center, and obviously to help the scientists thrive as well by creating this ecosystem for them?
Yeah, absolutely. I think in thinking about microbiome science at UCLA, we've really been able to think of a full, pipeline from the basic science to the translation, clinical work, and then even I think what Dr. Michelson will talk about is beyond that, commercializing and getting this really to the hands of the patient. And so in thinking about that, we're really able to identify where we may have gaps and then go for really targeted hires of excellence that will fill those gaps and really, dream team of people that will bring expertise along the entire path. And so that's how we identified Dr. Donaldson. We also have Dr. Gelsinger, who will be joining us later this year. And they've been doing really phenomenal work and truly understanding the microbiome on a basic level, but in a way that really applies to therapeutic development.
Amazing. So Greg, dovetailing on what Elaine just said, can you tell us a little bit about what attracted you to this position and what kind of unique research interests that you think that you bring to bear to the center, and how being in this environment has also been beneficial to you?
Yeah, sure, absolutely. So I'm relatively new here. I started at UCLA about a year and a half ago. The state of my lab, I think, reflects the answer to your question, which is I've been shocked how quickly we've been able to get our microbiome research off the ground. And that was enabled because when I started talking to Elaine about coming to UCLA and what she was building with you all in the Goodman-Luskin Microbiome Center was basically kind of like a playground for a microbiome scientist. So with the core facilities, being able to use technologies which are very complicated to set up and maintain, like germ-free animal models, but then also the connections to the clinicians, the gastroenterologists like Dr. Jacobs here. And so as soon as I got here, I had gastroenterologists saying, oh, do you need samples? Do you want biopsies? Do you need fecal samples? Sorry if you're not used to hearing about this, but
this is a big part of what we're going to talk about.
A lot of feces, yeah, I apologize, before lunch. But and that was something, as a basic scientist, it was always daunting and difficult to access those clinical samples. Even to communicate with a physician, to be honest with you, when I was a student, was an intimidating thing. And Elaine mentioned the sort of lack of egos at UCLA, and that was really apparent to me when I got here. Immediately I was welcomed as a colleague, And I hadn't felt that at I trained at some great institutions, don't get me wrong, I learned a lot from them. But this was a really great place to start sort of new work in this field.
Amazing. And can you tell the audience a little bit about your specific areas of interest as well?
Sure. So I'm a bacteriologist by training, and I'm an evangelist for bacteria. I think they have the potential to solve so many of our world's problems, not just health problems, but bioremediation, problems of pollution and climate change. It's a bacterial world. We live in it. The oxygen-rich atmosphere that we're enjoying all breathing today is maintained by plants, but was established by cyanobacteria billions of years ago. And throughout human and all animal evolution, we've lived with these bacteria in and on us, especially in our gut, that have such an intimate relationship with us. Some of these species or strains of bacteria will live with you for your whole life. And if you have a child, you will pass them on just as you pass on your genes. You will pass on some of these bacterial strains to your offspring. And what we study in my lab as bacteriologists is basically how these bacteria achieve this intimate relationship with their human hosts. This nutrient partnership that they engage in that has the potential to interface with basically all aspects of our health. I think we'll talk a little more about gastrointestinal cancers, and there's been an appreciation in the last 5 years that gut bacteria can influence the development and progression of colon cancer and also gastric cancer, which actually was known for quite a while before that. But fundamentally, actually, we already knew that these gut bacteria could interface with the pathways that control cancer development or the immune response. But it was a matter of getting sort of the right people together to work on this question, because you have immunologists over here maybe working on T cells, bacteriologists who historically were focused on pathogens, right? Everyone doing their own thing. So I think that's what things like this microbiome center are doing, is bringing us together.
Thank you, Greg. And Jonathan, you bring a kind of a unique perspective to the panel because you're a physician scientist. You also trained at UCLA. Can you tell the audience a little bit about your background and what prompted you to want to pursue microbiome science, and maybe even describe a little bit of the evolution of what you've seen at UCLA over the last few years. For those of you who don't know, like I said, Jonathan is a physician scientist, but he was also GI fellow at UCLA, as was I. And back when I joke, when I was a real doctor, he was actually a fellow with me on the inpatient service. And we scoped patients together, and he was amazing. I mean, you could always tell that Jonathan was brilliant, but also a very caring and responsible physician. So can you just tell a little bit about like how you fit into this picture, and then I'll ask you another question about translation.
Thank you so much for those kind words. I also look back fondly on those days. So I've been at UCLA since 2010, so I've been here longer than anywhere else in my life, more or less. And I think that speaks to kind of the support that I've received here for varying stages of my career, from being a fellow, learning how to do medicine, to science. I also officially received my PhD here. I previously trained in an immunology lab, but was kind of combining that with my MD. And then after becoming faculty, I've received incredible support and seen the emergence of the Goodman-Luskin Microbiome Center. So it's really been an amazing journey. And in terms of your question about what drew me to microbiome science, so this started fairly early, actually. When I was a medical student, I was studying immunology and model rheumatoid arthritis. But what became apparent was that the microbes are key to understanding the immune system. So that model that I was studying didn't actually develop without microbes present. And I became very interested in how microbes would control the immune system, particularly in the intestine. And so when I came to UCLA, I had a specific vision that I would be pursuing inflammatory bowel disease as my main clinical focus, but in particular trying to understand how gut bacteria would drive the immune system to lead to this kind of chronic inflammation, leading to all these terrible consequences for our patients. So I felt that by understanding the gut bacteria, we would be able to ultimately develop new approaches for improving outcomes for our patients, making them feel better, and preventing the need for surgery and other complications. And I've seen, of course, during the years here that the microbiome field has really grown. At the time I joined, it was kind of a GI-specific field within clinical medicine. But in the years that followed, people came to understand how important it was for our health, our immunity, and pretty much every discipline has become interested in the microbiome. And so I envisioned starting a lab that was just going to focus in on inflammatory bowel disease, work on our models, but I received all these requests to collaborate. And so quickly I changed my model, and so it became more of a collaborative model, which I worked with a lot of other investigators here. I still continue my own IBD-focused research, but I've become involved in a large number of studies with other disease states and helping to kind of bring understanding of the microbiome and the role of these various diseases and applying the concepts from gastroenterology and microbiome science to understanding of disease pathophysiology across a wide range of fields.
No, it's been remarkable to see this evolution. And, you know, what I remember back in the day when we were trying to get a biobank going and, you know, you were going through all the processes because we didn't really have an infrastructure set up for that. And you played a huge role in establishing a process so that doctors like Dr. Donaldson could come in and say, oh, okay, I have samples. But can you talk a little bit about the Goodman-Luskin Microbiome Center and how translation is facilitated, you know, beyond just people doing their individual research in basic science labs?
Yeah, so the establishment of the Goodman-Luskin Microbiome Center really made a huge difference in being able to transition from having ideas and plans and kind of early pilots. You know, people started their own collections, but in order to make this systematic and make it accessible to our basic science colleagues and other investigators, we really needed to organize it into these cores. And that's what the Microbiome Center, in part, has contributed to the community, that we create these organized resources where investigators could deposit their samples. They could be archived in a safe manner with backup systems and with software that allows us to track where everything is located, so that if someone comes in, they need specific stool samples from a particular type of patient, then we can find them right away, get them the samples, and they can do their work. And so everything became centralized, scaled up, and made more accessible to our colleagues. And so it's no longer as intimidating for a basic scientist to say, well, I want to look at some stool samples from a particular patient population. Well, now they just know who to go to. There's a biorepository, and they can get those resources.
Yeah, and I think, you know, basically what you all three of you have kind of described is like laying the railroad track down for what now Dr. Michelson has enabled us to do at UCLA with the partnership between the California Institute for Immunology and Immunotherapy and the Goodman-Luskin Microbiome Center. And Gary, you know, we could spend the whole panel talking about you. Honestly, for those of you who don't know, Dr. Michelson's an orthopedic surgeon, but a prolific entrepreneur, and also somebody who's been very generous in his lifetime supporting science. And before we talk about the microbiome vaccine project and the microbiome work, can you talk a little bit about your vision for the institute, which just for speed, I'll call it CIII. But when we first started talking about CIII, you know, many years ago now, you always wanted to establish a center of excellence around immunology and obviously harnessing the power of the immune system. Can you tell us why you chose immunology and kind of the rest of us have been along for the ride, but you've been our fearless leader. So maybe just to explain why immunology as the focus.
Yes. So I was an orthopedic surgeon, and they're basically carpenters. And if you know you're going to be an orthopedic surgeon, you don't go to liver or lung, or you skip all those. Okay. So it was really strange that at some point I became interested in our immune system. This is about a decade ago. And I guess what surprised me was it seemed to be missing in this discussion of all of the chronic diseases that afflict and kill all of us. So whether it's heart disease or it's cancer or it's Alzheimer's, the big 5 chronic diseases that kill us all are all mediated by our own immune systems. And then I had the pleasure of having dinner with Liz Blackburn, who won a Nobel Prize on her work on telomeres and aging. And I walked away from that. She said, look, you're born, you develop for about 20 years. And everything that happens after that is an attack by your immune system. It's chronic inflammation that builds up and it destroys your tissues. So that was pretty startling. And as I looked around, it became apparent to me there were 2 great frontiers in medicine. So up until that point, every disease had its own sort of research program. And they would just keep drilling down, trying to get to what it was the answer. But there seemed to be two big answers to everything. 8,000 diseases are potentially amenable to gene editing, and all these chronic diseases are mediated by our own immune systems. So immunology seemed to be essential.
And obviously we went down the road of establishing the institute, and from the very beginning we talked about key pillars. Obviously cancer and immunotherapy is always going to be a big one, and we've been talking about that from the beginning. But also from the beginning you were always focused on the microbiome. Every time we drew a schematic, and for those of you in the room who have been part of this journey, even we started on this before the pandemic, and that gave us a little bit of pause while we were all just trying to stay alive. But as we rebooted and came out of the pandemic, again, you were very focused on microbiome and immunology from the beginning. So can you tell us a little bit about why you think it's so important? And then we'll also ask the panelists about what they're working on.
Yes. So when you're in medical school, they teach you that when the patient says, doctor, what's causing my disease? You never say, I don't know. You go, well, that's idiopathic. Unless they go home and look up that word, which means I don't know. But anyway, it's so interesting because when I was in medical school, we did not know what caused a great number of diseases. And Elaine and I were just chatting about this. So it turns out that rheumatoid arthritis is caused by a bacteria in your large bowel that never even infected you. But our T cells are rushing down to that endothelial lining to our gut every time we eat a meal. It's a 5-alarm fire. Insulin goes, woo, 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 and 80% of your immune system's down in your gut. And what happens is they sample a bacteria, basically the antigen on the surface of the bacteria, and then they turn around and mistake one of your tissues, in this case your synovial lining, for that bacteria and start attacking your own tissues. So here you have an organism that's minding its own business, really, in your gut, and it's causing horrific disease. No, your immune system's causing horrific disease, I guess would be a better way to put it. And so now what we're learning is, and we've known this for a while, we just didn't have the AI engine to sort through all the big data, but what we knew is you can give a CAR T to a group of patients. 40% are cured with a single injection. 60% it acts like you never gave them anything. Now, if you give the 60% antibiotics and wipe out their microbiome and take a fecal transplant from somebody who was a responder, a significant portion of the people who are non-responders now become responders. It's quite simple. Your own microbiome, or in this case, a borrowed one, is controlling your response to chemotherapy. And we see this in a number of places. And as was already pointed out, I think by Greg, these bacteria have been co-evolving with us for as long as we've been mammals. And some of them seem to just be getting along with us. We give them what they want, they give us something, but some of them are clearly superstars, and some of them are really bad. So it's just a fertile area.
And Elaine, when you started hearing obviously about CIII and Gary and his focus about the microbiome as part of our initial foray into actual scientific projects as part of CIII. Can you talk a little bit about how the MVP kind of idea came to be, and then we'll ask also Jonathan and Greg about what they're working on specifically as part of the project.
Yeah, absolutely. So the MVP, the microbiome vaccine program that we have going on, and we just recently started, is really an effort to harness the microbiome to decrease risk for gastric and colorectal cancers. I think, as was mentioned, there's been a great advance in studying the microbiome in cancers, but most of the focus has really been on response to immunotherapy, which is very important. But on the basic science side, there's this huge influx of findings showing that the microbiome can also influence early risk in cancer initiation and tumorigenesis. And so we'd like to really tackle that issue. with innovative approaches. As I was mentioning earlier about just the collegiality and the ecosystem of diverse expertise at UCLA all at the same place, that's truly the approach that we're taking for this program in bringing together structural biologists, computational scientists, microbiologists, physicians and gastroenterology and immunologists. And so this is really a multidisciplinary effort that's tackling this problem or aims to tackle the problem from multiple angles with core pillars.
And Greg, in terms of your involvement, can you describe a little bit about what role you play and what's of interest to you as part of this MVP project?
Yeah, sure. So I never, to be honest, never thought I would work on cancer biology or immunology as sort of a bacteriologist. But in studying these gut bacteria, what became clear to us in thinking about what enabled this long-term sort of co-evolved partnership we've been talking to is that their interaction with immune cells, but especially the epithelial cells that line your gut, is really complex and goes beyond this sort of antagonistic relationship that has been studied in the context of pathogenesis, like enterotoxigenic E. coli or salmonella that gives you gastroenteritis, or these certain bacteria that we know can sort of induce tumorigenesis. And so these cancers of the GI tract, vast majority, they're epithelial. So it is these epithelial cells that become the tumor. These epithelial cells have such a tough job, right? Because they absorb the nutrients, that's how we feed ourselves, and they're exposed to the environment, all these toxins, whatever we're taking in, and they interact with these bacteria. But what we've had already been looking at in the lab is that in addition to these antagonistic interactions, there are bacteria that can induce the opposite of a tumorigenic sort of differentiation pathway for the epithelial cells. So what we're really trying to do is see if we can take advantage of this in a way where we can manipulate the microbiome to prevent sort of polyps or very early stage cancers from forming. Can you have a probiotic or microbiome-based intervention that prevents the emergence of a tumor entirely? So it's like a true sort of prophylactic against cancer, in addition to something that could reverse sort of dysplasia that you already have.
And Jonathan, I think for maybe people in the audience, can you describe kind of the concept of looking at vaccines versus potentially competing bacteria, pathobiont, the approach that you've all laid out in terms of how we might be able to prevent cancer? Because vaccination is one way potentially, and then there may be other ways as well, correct?
Yeah, so as Greg mentioned, there's a number of characterized bacteria now which have been found to be able to induce cancer. And this is a potentially major driver, particularly of earlier onset of cancer. There's emerging evidence of that. And so one of our goals has been to establish defense against these kind of cancer-promoting microbes. And there's different ways of doing that. One is to invoke the immune system, a more traditional vaccine. But another idea is to use our microbiome as an initial barrier and defense against these cancer-promoting microbes. And so not everyone's going to be colonized with these cancer-inducing bacteria. They have to establish colonization, they have to outcompete the other microbes around them, and they have to be able to interact with these epithelial cells lining the intestines and the GI tract in order to induce cancer. So if you can find the right combination of bacteria that can prevent those cancer-inducing bacteria from getting a foothold, you can, through what has been called colonization resistance, you'd be able to provide defense against development of cancer caused by these microbes. And so that's the fundamental concept. And our approach has been to try to discover new microbes that are poorly characterized or not even characterized at all, perhaps known only at the level of DNA, and to find combinations that allow for new defense strategies to prevent these cancer-inducing microbes from getting a foothold and then from having their effect on the epithelial cells.
Yeah, I appreciate that because I thought that was one of the most fascinating approaches to the proposal that you put together, was that, of course, vaccination is one strategy, but what if there's a strategy of building kind of a force field that prevents cancer, utilizing the microbiome? And Gary, as we kind of close out this session, you've talked a lot about how you've always wanted, as we build out the CIII, to bring scientists together from different disciplines to kind of cut down barriers. Can you say a little bit about what this, based on what you've seen elsewhere, how this vision is different, and why you think that it'll be successful as we put it all together.
Well, first, to your credit and to Elaine's, UCLA just had a remarkably deep and broad bench. And we all know the story about the 5 blind men who encountered the elephant. One grabs the tail and says it's a skinny snake. The other one's got the trunk and they go, no, it's a huge snake. Somebody's got a leg and go, no, it's a tree trunk. Somebody else, you know the story. Anyway, the point of it is, these are daunting challenges. And we're very fortunate that we actually have the means to process the massive amount of data to start to derive meaning from this. But at the end of the day, you need a multidisciplinary approach to deal with these. These are not one-brain problems. I think her team might have 10 different people on it. And at the end, they're going to have to start having engineers. It just requires that kind of team to address these very, very large and challenging problems.
Well, I appreciate your generosity, Gary, and your leadership, because you've really motivated my family and many of the other founders to jump on board. And I also wanted to thank Elaine, Greg, and Jonathan for not only being outstanding scientists, but for proving the concept that we could start a CIII right here with some of the faculty that already existed at UCLA. And as we recruit more faculty, you mentioned Diego Gelsinger, and as we build out the laboratories at the Research Park, I think we are going to finally be able to fulfill the potential of everything that Dr. Michelson and others have been working on all these years. So I wanted to thank you. Thank you for participating. Thank you all for your attention. And thank you. And outside, you'll also find some information about the California Institute. You should get on the mailing list, because we'll be having a lot of news in the coming months and years. Thank you very much.
Hi everyone, my name is Tibor Anak. I'm a business development officer at UCLA TDG. I would like first to thank our panelists from the microbiome panel, and now I'd like to introduce one of our longtime originating sponsor of LABEST, Kite, a Gilead company. Joining us from Kite is Kevin Cognetti, Vice President Corporate Strategy. With nearly 15 years of experience in the pharmaceutical industry, Kevin is responsible for shaping the growth path forward at Kite by integrating the business, financial, scientific, and strategic analysis into one strategy connecting investment, resources, and plans to the ultimate vision and mission of Kite. Most recently at Kite, he shepherded the incorporation of Kite's in vivo and multiple myeloma business acquisition into Kite, as well as working closely with the business to unlock accessibility to CAR-T therapies across the globe. Kevin received his bachelor degree, JD, and MBA from Georgetown University. Kevin, welcome to LABEST.
Thank you, Thibault. Thank you, LABEST. Thank you, UCLA. I'm going to spend a few minutes and talk about where Kite started here at this very spot at UCLA, and then talk about what we have achieved, where we're going, and then close with a video that will bring it all to life. It all starts with our mission. And our mission, under the leadership of Dr. Beldegren, started about 10 years ago in bringing CAR-Ts and delivering them to patients for a potential cure. Patients cure CAR-T. That is what we do every day, and that is where we derive our work and our progress. And as I said, it all started here in California, here at UCLA, and in LA. And we've expanded upon that. And we've done that in about half our employees are still California-based. We have 5 centers here in LA, including a major research center and a major manufacturing center and our headquarters. And we also continue to partner with the community here, both in sustainability as well as to move CAR-Ts forward. And from that start, we have achieved a lot. We have now reached over 30,000 patients with CAR-Ts and giving them potentially more time with their families, with their friends, and in their lives. We've done that across 2 therapies, 5 indications in over 30 countries. And underneath all of it, it's this buoy of research and manufacturing that has been built out here and in other places to really drive across that 1 million square feet to make and deliver those CAR Ts. But it's not good enough, and we have work to do. Currently, only 2 out of 10 patients that are eligible for CAR T gain access and receive it in the United States. Only 3 out of 10 in other parts of the globe. That leaves a lot of work to do. And it's not enough for Kite to be doing it, but it's a community effort. It's an industry effort. It's governmental effort. But we're going to do our part, and there's 3 ways in which we're doing it. First, connecting patients to care. What we find right now is that 2 out of 10, that numerator of 2, is really held back by sites of care and by the science itself. And so within the sites of care, we need to move closer to the patients. The patients struggle to get to the distance, the logistics, the proximity. Patients need a caregiver. The economic burden of that often makes them choose different therapies with less generous outcomes. And therefore, we, as a group, are working across industry to expand the sites of care to community oncologists and to break down some of those barriers. But we also need to change the science and improve the science. So many of the CAR Ts today have a safety profile that limits the number of patients that get it. And so at Kite, we have 2 late-stage development programs that target just that, bring safer products to market and increase people's chances for a cure. We need to move forward and change the denominator, the 10. That 10 is only in hematological cancers today. And so what we look to do is change that and bring it to more patients in more places with more malignancies. And we're doing that in autoimmune disease and in solid tumors. And with that comes the heterogeneity problem that we face, and our new therapies include armoring and dual targeting. But that's still not enough. And so we're investing in disruptive technologies. As you heard earlier from Eli Lilly, we've also invested and are starting to develop a in vivo CAR T treatment. Bring the outcomes within CAR T to patients in a more convenient and easier way. And so with these 3 barriers overcome, we believe we can change the growth trajectory of CAR T while also providing more and more patients a chance at time, a chance at a cure. As I said before, it's all buoyed on the expertise and the manufacturing abilities that are required to do such a inundated and involved treatment, whether it be logistics of chain of custody, the precision in the machinery, the precision in caring for individual cells. We at Kite focus on this. We've invested in this in California and across the globe. And we've done that to great success. But rather than me go through this, I want to share with you a video from a patient in our leadership that shares why this is so important. I thank you all for your time, and let's cue the video.
Cures only matter if you can reach the patient on time, and that's where manufacturing comes in. CAR T-cell therapy is a one-time treatment made from the patient's own white blood cells, and it's potentially curative. And so when patients and physicians feel as if they've run out of options, this presents a possibility for those patients.
I had already been through 2 completely failed chemo lines. When my doctor said we're moving forward with CAR T, I really didn't know what that was. I think patients often don't understand what goes into the process or the manufacturing.
We are not pulling vials or syringes off a shelf. We're actually taking a patient's cells and delivering a personalized therapy back to them.
Because of the nature of CAR T cell therapy, manufacturing isn't just the operations, it's the product itself. Time is critical because these patients have very aggressive cancers and every moment matters.
Am I going to survive? Am I going to live long enough to receive it? Those thoughts go through your head, especially when you're fighting cancer.
Delays are not only inconvenient, but it also can determine whether or not a patient is still eligible for treatment.
Physicians are trusting Kite with their patients' cells, so delivering these therapies reliably with a quick turnaround time, with a high success rate, this is part of the overall outcome and experience that the patient and provider have.
The care coordinator met with me and she kind of went over the CAR T process. You're going to go through apheresis, and then you're going to receive your cells. It just didn't register in my mind as a patient that this is only one treatment.
We need to ensure that the way we are manufacturing, the turnaround time is really aligned with the expectation at an hospital level. And that's what will make CAR T scalable.
We know cancer doesn't take a break on weekends or on holidays. That's why it's critically important manufacturing remains open 24 hours a day, 7 days a week, 365 days a year.
There's no shortage of innovation in this space, but proving you can deliver at scale reliably is more rare.
There's such an amazing science behind every step of the way. It's just so careful. It meant the world to me and to my family. CAR T is here, and it's changing the world of cancer treatment.